Hello guys, welcome to the knowledge domain, the concept of life sciences and biotechnology. Today we are going to learn the hypersensitivity and we will focus on to a slightly more detail on the type 1 type of the hypersensitivity. So the immune responses are associated with the multiple strategies to reduce damage to cell by turning off responses. When pathogen is clear, and avoiding reactions to the self antigen so that we are protected. There are checks and balances. Those are breakdown results in leading to the immune mediated reactions. Those are more detrimental than the protect. Some immune mediated disorders are caused by failure to the immune tolerance. Autoimmune disorders are caused by an inappropriately vigorous, innate, and or adaptive response to antigen that pose little or the no threat are called the hypersensitivity reaction. Based on the allergen, the protein molecule or the molecule that is responsible for allergic reaction or the hypersensitivity reaction, and the time and the duration in which there is the symptoms manifestation. This based on this duration, it is can be divided into the immediate hypersensitivity reaction, where the symptoms manifestation it is happening within a very short time period after the immune stimulus. Whereas in case of the delayed type of the hypersensitive reactions, it takes hours and sometimes it takes days. There are major histocompatibility complex, the short form of the MSCR of three types 1, 2, and 3, and these are the molecules that are responsible for differentiation of self from the non-self. So here we are going to discuss our class 1 MSC molecule and the class 2 MSC molecule structure. So the class 1 molecule and class 2 both are made up of the alpha and the beta molecules. In case of the class 1, the alpha molecule is having alpha 1, alpha 2 and the alpha 3, three domains. And the beta 2 microglobulin is having just one domain. And in case of the class 2 MSC molecule, alpha 1 is having 2 and the beta molecule is also having the 2 domain. So it is a both are heterodimers. So where the allergen binds in case of the class 1 and in case of the class 2 molecule. So in case of the class 1, this is the cleft, the peptide binding cleft that is present between the domain of the alpha 1 and the alpha 2 of the class 1 MSC molecules. Whereas the cleft is between the alpha 1 and the beta 1 of class 2 molecule. There are the four types of the hypersensitive reaction based on the immune mediators and the type of the initiation it is happening. So in case of the type 1, the initiation is because of the allergy and the atopy, or it is because of the all of them are because of the allergen, but here it is allergy and the atopy and the mediator is the IgE. So here you can see this is the allergen. Allergen is a molecule that initiates the allergic reaction. It is binding with the antibody that is the IgE. The immune mediator is the IgE. Then this is going to activate the granulated cell like the basophil and the mast cell. And after that there is a degranulation. So the immune mediator is the IgE. The mechanism involved in the allergen induces cross-linking of the IgE bound to mast cells or basophil and the release of vasoactive mediators which are responsible for various vascular activities like the vasodilation. And where do we see the type 1 hypersensitive reaction? It is like the systemic anaphylaxis, localized anaphylaxis, such as the hay fever, asthma, hives, food allergies, and the eczema. Then coming to the type 2 hypersensitive reaction, it is initiated with the antibody. You see, antibody mediated hypersensitivity. So antibody is binding to the antigen. This is the target cell that has to be eliminated, has to be killed. Okay. So what are the immune mediator IgG and the IgM? So it can be IgG, it can be an IgM. So antibody directed against the cell surface. This is the antigen, that is the cell surface. By the complement activation, you see the C3b is binding here, that leads to the complement activation, or it can be killed by the cytotoxic T cell, the ADCC, antibody dependent cellular cytotoxicity. Where do we see the type 2 hypersensitivity reaction in case of the blood transfusion? So we know the antigen A, antigen B, RS positive, these are the antigen molecules present on the red blood cells. So usually when the red blood cells are involved, we see the type 2 type of the hypersensitivity reaction. Example is the blood transfusion, erythroblastosis fetalis. This is the disease when the RS negative mother is carrying RS positive child. The first child delivery is okay, but in case of the second child delivery, there are the complications. So that is also involved the RS positive of the red blood cells. Then there is the autoimmune hemolytic anemia. Then coming to the type 3 hypersensitive reaction, it is initiated by the antigen and the antibody complex. So this is the immune complex mediated hypersensitive reaction. Initiation is because the immune complex, the mechanism, this is the antigen antibody complex deposited in the various tissue, induces complement activation. You see the C3b, the initiator complement, and an ensuing inflammatory response because of there is a huge migration of the neutrophils. Where do we see the type 3 type of the hypersensitive reaction like the Arthur's reaction, serum sickness, necrotizing vasculitis, then glomerulonephritis, rheumatoid arthritis, systemic lupus erythematosus. So here the systemic lupus we have discussed, rheumatoid discussed, multiple sclerosis discussed. But there is a drug reaction like the allergy to penicillin and the sulfonamide. They are the type 3 type of the hypersensitive reaction. Then infectious diseases like the post-streptococcal glomerulonephritis, meningitis, hepatitis, mononucleosis, malaria, and the trypanosomiasis. Then coming to the type 4, this is the delayed type of the hypersensitive reaction. In this case, the immune mediator is the T cell. This is the T cell, the sensitized T cell. So they are secreting cytokines are the molecule. They are the language between the cells. So the cell they talk to each other through the cytokines. So the T cell is talking to the other cells through the secretion of the cytokines. What are the T cell involved in this case? The Th1 and the Th2 cytokine that activate macrophages or the cytotoxic T cell. So it is saying to the other cell about the direct to cause the cellular damage. Where do we see this one? Dermatitis, tubular lesions, and a graft rejection. We see the delayed type of the hypersensitive reaction. There are the intracellular bacteria, those are involved in the type 4 type of the hypersensitive reaction, as the Mycobacterium tuberculosis, Mycobacterium leprae, Brucella abortus, Listeria monocytogenes, then pneumocytosis, Candida albicans, Histoplasma Capsulatum, Cryptococcus neoformans, then the intercellular parasites, the protozoans, Leishmania species, then viruses, herpes simplex virus, variola, that is the smallpox cause, measles virus, then there are the contact antigens like the picri chloride, hair dyes, nickel salts, poison ivy, and the poison oak. Then coming to the allergen, that those are involved in commonly for the type 1 hypersensitive reactions for the pollen, plant pollen results in the ryegrass, ragweed, timothy grass, beech trees. The drugs, we already know penicillin sulfonamide, they are the local anesthetics and the salicylates. They are the mold spores, animal hairs and dander, latex, foreign serum, vaccines also results in the type 1 hypersensitive reactions. Then the food, some people are very allergic to nuts, seafood, egg, peas, and the beans and the milk. The insect product result in the type 1 type of the hypersensitive reaction, the bees and the venom, wasp venom, ant venom, cockroach calyx, and the dust mites. Then the principal mediators of the type 1 hypersensitive reactions, the histamines we have already studied earlier, histamine, the heparin, they increase the cellular permeability, the vascular permeability, and the smooth muscle contraction. The serotonin also for the smooth muscle contraction, and the increased vascular permeability. Then the eosinophil chemotactic factor and the neutrophil chemotactic factor are responsible for the eosinophil and the neutrophil chemotaxis. Then the proteases, they are responsible for the bronchial mucus secretion, degradation blood vessel, basement membrane, and generation of the complement split products. Then for the secondary effect, the platelet direct growth factor leads to the platelet aggregation and degranulation, contraction of the pulmonary smooth muscles. And the leukotrienes, the slow reactive substance of the anaphylaxis, SRSA, this increase the vascular permeability, contraction of the pulmonary smooth muscles. Then the prostaglandin, they result in a vasodilation, contraction of the pulmonary smooth muscles, and the platelet aggregation. Bradykinin, they increase the vascular permeability and the smooth muscle contraction. Cytokines like the IL-1 and the TNF alpha, they result in the systemic anaphylaxis, increased expression of the aerosol molecule in the vascular endothelial cells. IL-4 and the IL-13 increase IgE production. Then IL-3, IL-5, IL-6, IL-10, TGF-beta and the GM-CSF, they result in various effects.